students, in today's class we are going to discuss about the cross conducted by Gregor Johann Mendel. He had conducted two kinds of crosses, monohybrid and dihybrid cross. And in monohybrid cross, the characters were considered as one at a time and two alternate forms were crossed. But in dihybrid cross, two distinct characters were analyzed for the experiment. So today we will discuss about the monohybrid cross. The character he had chosen was the height of the plant. You can see two plants here, two pea plants. One is a tall plant, the other is a short plant. And the experiment he had conducted by crossing the tall and dwarf plant. When you take crossing the tall and dwarf plant means it is same as that of cross-pollination, controlled cross-pollination. As we all know, when cross-pollination occurs, there is a transfer of pollen grains from the flowers of the plant, from one plant to the other plant. That is why we call it as cross-pollination. So when we take cross-pollination, naturally, we can't wait for a wind to blow or a honeybee to visit, and then we have to wait for the honeybee when it goes to the other plant. So there is a kind of artificial pollination, cross-pollination or controlled cross-pollination. Let us see what exactly a scientist can do for this controlled cross-pollination. You can see two flowers. Imagine this flower represents the flower of the tall plant and this flower represents the flower of the short-heighted or dwarf plant. Now we are going to make an artificial pollination by the help of few equipments. We need a scissors, then a brush, a scissors, brush and a polythene bag. Let us see how it can be done. First of all, by the help of a scissors, you are cutting the anthers. You can see that anthers are arranged in black color, anthers, which will provide the pollen grains. So you cut and thrown all the anthers. Suppose this flower, the anthers are cut and thrown. Alright. And now here only stigma is available. And by the help of a brush, you can dust the pollen grains of this flower. By the help of a brush, you can just dust the pollen grains from this flower and take that brush to the stigma of the second flower. So what you can expect, suppose the pollen grains are present in this way and by the help of a brush you have dusted to this stigma. So many pollen grains are sticked on the stigma. Now we have to preserve that no other contamination should happen. So if this plant should not be contaminated means you have to cover it with a polythene bag. If you don't cover, what will happen is that from through the wind or by any other agency, there is a chance of the pollen grains to come and stick here. So what exactly happens is that the pollen grains will grow and it will be fertilized at this ovary, right, with the ovule, and you can see that one plant will be one seed will be grown there. And for example, these are the pods with the seeds. These seeds are grown and as a result plants can be grown by sowing these seeds. This is what we call for the controlled pollination. Now Mendel had taken self-pollination also. Self-pollination, we do not have to do anything. It will be pollinated by itself, because the pollen grains position and the stigma position both are side by side. So automatically it can fall on the stigma, or even by any other agencies also it will be pollinated. So you can see that cross-pollination and self-pollination is done in Mendel's monohybrid cross. Now let us see what are the different steps taken by Gregor Johann Mendel for his monohybrid cross. You can see in this white board we have taken few steps. There are 2 steps in Mendel's monohybrid experiment. 2 steps. We have already learned what are genes, what are alleles, then comes recessive, then dominant, homozygous, heterozygous. By keeping all these terminologies here, we are going to see how Mendel had done this monohybrid cross. You can see step number 1. A tall plant, the one which I had taught just now. A tall plant with homozygous capital TT, that means the alleles are homozygous tall, is cross-pollinated, the procedure which we have seen, with a homozygous dwarf plant. So here both parents, male parent will contribute the pollen grains, and the female parent will contribute the ovules. So here you can write gametes. The first step will be parental genotype. Second is the gametes. Gametes are always written in a circle. So here 2 gametes are produced, because one allele will come to one gamete, the other allele will come to the second gamete. Here also the same thing. The first allele will come to the first ovule, and the second allele will come to the second ovule. So we have got 2 pollen grains and 2 ovules. Now we are going for the fertilization. Now let us see all the chances or the probabilities. Chances. You can't have exactly say that this allele of the pollen grain will cross with this. No. So you can say that the capital letter T represent the first pollen grain. If this pollen grain will fertilize with this ovule, what can happen? Both will develop into a gamete capital T, small t. Then second chance, the same pollen grain can have an option to the second ovule. Then the second pollen grain can have a chance to the first ovule. Then again the second pollen grain can have a chance to the second ovule. That means all 4 probabilities or chances we are taking, because we do not know which pollen grain will fertilize with which ovule. There is all happen inside the flower or inside the pollen tube, what we have studied in fertilization in reproduction. So when these first option is chance is fertilizing, what can happen? Capital T small t. Second, the same thing, capital T small t. Third, capital T small t. Fourth, capital T small t. All plants are heterozygous tall, because capital T is dominating over the small t. So all 4 plants will be heterozygous. So you can see that. See, when this fertilization happens, the pod will develop. The entire pea pod will develop. As a result, 4 seeds are produced. These 4 seeds will be developing into 4 plants. All are tall. Now in the second step, Mendel had taken the same generation, same plants which was formed in the first step, and all these plants are called first generation plant, F1. F stands for filial. First generation plants, F1 plants, all were tall. In the second step, what he had done is that he took two F1 plants and self-pollinated. He did not go for cross-pollination, self-pollinated. Look at that. The first parent, capital T small t, because capital T small t, and the second parent, it can be this plant. So two plants are heterozygous tall. And again, what is the next step? Parents we have got. Now we have to trace the gametes. This is a male parent. So two pollen grains are produced. One with first allele capital T, second with second allele small t. Here also the same thing, capital T, small t, and see all the chances or possibilities, just like the crosses we have done, fertilization. This allele fertilized with the first ovule allele, capital T, capital T. Then comes capital T, small t. Then comes capital T, small t. Then comes small t, small t. Fourth probability, fourth option. So from this you can understand that second generation, F2 generation is totally different. The F1 generation all were tall, heterozygous tall. But look here, the first plant is homozygous tall. Second and third plants are heterozygous tall. And the fourth plant is a dwarf plant. So entirely different results we are getting. We can get the plants, resulting plants, F2 generation plants ratio with that height. First is tall, second is tall, third is tall. So three tall plants. And the fourth one is a dwarf. So our ratio is 3:1. So when you look at those four plants, 3 were grown tall and 1 is grown dwarf. So 3:1. Now when you looked at these 3 plants, you could see that there is a difference. The first tall is homozygous tall. Second and third are heterozygous tall. From this you can understand that there is a difference. So we have to show that also. Allelic form we can show. So when you trace out, one is perfect homozygous tall, second and third are heterozygous. So we can again split this 3 into 1:2, 2:2, the last dwarf 1. So 1:2, 2:1, we have modified the ratio. This ratio 3:1 is the ratio which you have got from the physical appearance, observable character. So this character is called phenotypic ratio or phenotypic character. So phenotypic ratio is 3:1. But when you go more analysis, allelic constitution or gene constitution, you have got genotypic ratio 1:2, 2:1. So we have got two types of ratios. One, phenotypic ratio with physical appearance, that is 3:1. Then comes the genotypic ratio 1:2, 2:1. So from this you have understood that F1 generation all were heterozygous tall. F2 generation 3 were tall and 1 is dwarf. Right? So out of the 3, 2 are heterozygous tall and 1 is pure homozygous tall. And the last one, there is no hetero or homo, that is only the dwarf small t, small t. So again the ratio 3:1, that is a phenotypic ratio, external appearance ratio. 1:2, 2:1 is the allelic composition, that is genotypic ratio. So from this you could understand that the first grandparents were homozygous. They got heterozygous tall. Then when they have crossed, these F1 heterozygous tall plants were crossed, a new character has originated, which was their grandparent. So, here you are getting both the steps. I hope that the monohybrid cross is very clear for you. Okay then, thank you.